So as we begin to build our biologically important molecules, we're going to continue to talk about this building up. We talked about condensation before, putting monomers together, liberating a molecule of water. We're going to expand on that because when we talk about building up or tearing down, it really does fall under the heading of metabolism. Now that condensation reaction, it's going to use enzymes. Enzymes are catalysts for chemical reactions. What that really means is they allow chemical reactions to occur faster and more easily. Enzymes will never facilitate a reaction that will not naturally occur in nature. It just doesn't happen. But additionally, with enzymes, you have to use energy. And what's our form of energy in our bodies? ATP. That's our storage form of energy. So with enzymes and using energy, we take our monomers, amino acids for instance. We assemble them with a condensation reaction into a polymer, which we call a polypeptide or a protein. That's just one of our examples. So our building up, condensation. The opposite, the tearing down, the breaking off of a monomer from a polymer, hydrolysis. What does that word literally mean? Hydro, water, lysis, to break. So if you know one of these reactions, then you already know the other because it's just the opposite. Taking a molecule of water and an enzyme, breaking the water into hydrogen and a hydroxyl group to break those apart. You see how that works? So again, condensation reaction, which we've already seen. We take our monomers with enzymes. We're going to remove a hydroxyl. You remember that from last chapter. Our hydroxyl group from one monomer, a hydrogen from the other. The enzyme's going to put that together to form water, which we eject. Then we're going to link those monomers together and form a polymer. Sometimes it's called dehydration. Do you see where they get that? Condensation, dehydration. We're basically forming water. In our illustration, we have some carbohydrates, monosaccharides. We use enzymes, liberate our water molecules, and now we have these covalent oxygen linkages that are holding our monomers together into our growing chain of carbohydrates. Now, the only reason you know this is a condensation reaction and not a hydrolysis reaction is because of the direction of that arrowhead. Right? Because if the arrowhead was pointing in this direction, that would be hydrolysis. So do you see, if you learn one and just reverse the arrowheads, you know both already. See how easy that is? But this is this generic condensation reaction, taking monomers, putting them together into polymers. We'll get a little more specific in detail, especially with our proteins. So hydrolysis is the opposite. And there's really no need to make this any harder than it is. You just reverse the arrows. Using different sets of enzymes, taking our polymer, adding water and energy to separate those O linkages, and now we have our individual monomers. So lysis is going to be the key. Lysis is to break. We're breaking the polymer into monomers. If you can hang on, bless you, if you can hang on to that definition, you will know that the other reaction is the building up, hence condensation or dehydration. Does that make sense? Because really, condensation, dehydration, in your mind, you just have to memorize that it's the building up, whereas lysis, it's built into the name. Lysis is to break apart. So that's one easy way you can remember both of them, and just by remembering that one word, lysis, in hydrolysis. So now let's again get into the weeds here, get into some specifics, and we're going to start with proteins. What do proteins do? What do proteins not do? That's probably the shorter list. Proteins do everything. You got them, enzymes. Not all enzymes are proteins. You can have some ribozymes made out of RNA. Hormones, some of those are proteins. Transport. Yeah, proteins have such diverse functions in our bodies. So many. Just a list, you don't have to remember this. This is just to give you an appreciation of everything that proteins do. Again, as I mentioned, I think it's easier to try to find something that proteins don't do. So, monomers, we're again coming back to our condensation here. Monomers of proteins are amino acids. And they're 20 different amino acids. And we're going to learn our amino acids and their properties. Now, when we look at the basic amino acid, this is the one that we sort of saw as our stereoisomer. You didn't realize that's what you were looking at, but that was our amino acid. Because there are 4 groups. All right, so our basic structure of our amino acid, at the core we have a carbon. Call it the alpha carbon. We're going to have a carboxyl group. Remember our corn? It's a carboxyl group. There's our amine group. We're going to have a hydrogen stuck on there. And then this R group, why they decided to call it R, I don't know, because it's the variable group. That's the one that's really going to define which of our 20 amino acids we're talking about. If we just knew all these other parts, they would all look the same. So our R group, this variable group, that's going to be the part that gives the characteristics to a particular amino acid. Thank you. They'll probably be back. Maybe we'll be done by then. So it's the R group that's going to dictate whether an amino acid is negative or positive, whether it's polar or whether it's hydrophobic. We can have amino acids that are hydrophobic. So if amino acid is hydrophobic, what could you already predict is going to make up that R group? If the R group determines the property, and I'm telling you this amino acid is hydrophobic, what can you already assume about that R group? What's it composed of? Hydrocarbons. Hydrocarbons, exactly. And the number of carbons is going to determine which amino acid it is. So you can have such a huge diversity of these amino acids. Once again, we have stereoisomers, but the L or S, depending on which you're using, the left-handed stereoisomer, that's the one we're typically going to find in our proteins in nature. Now, here's a new term. Remember we used the term amphipathic? What does amphipathic mean? Both. It had different parts. Our phospholipid is amphipathic. It had a hydrophilic portion and a hydrophobic portion. Well, now here's a new word, aliphatic. Aliphatic sounds a lot like amphipathic, but they're different. This word, aliphatic, really refers to hydrophobic. And I would prefer to use hydrophobic. I just want you to be familiar with it. And as we look at several of our amino acids, notice we have this R group. And this R group in this particular illustration adds another carbon. Add some more carbons, add some more carbons, add some more carbons. So with glycine, our R group is simply a hydrogen. It's really not going to be much of anything, but it's not charged, so they say, okay, we'll stick it as hydrophobic. But it's very little. And as you add more and more carbons to the chain, what's happening? It's becoming more and more hydrophobic. The longer the hydrocarbon chain, the more it can disrupt water, the more hydrophobic it becomes. And so that's one way in which we define the properties of our particular amino acids based on that R group. So now let's look at how do we organize and categorize our 20 amino acids. So first, we've got this group, and notice we've got glycine. Glycine's going to appear in multiple groups, just because glycine is so simplistic. Because there's our R group of glycine, just a hydrogen. That's it. So this first group are the non-polar amino acids, and again, said to be hydrophobic. Now, this is not a chemistry class. I am not going to expect you to draw structures. You'll have to do that in biochemistry, I'm sure, but here you won't. You will have to know the name, the 3-letter, and the 1-letter abbreviation for all 20 amino acids. You need to know the name and the category of the amino acids, plus the 3-letter and the 1-letter abbreviation for each of our 20 amino acids. Sorry? Yes. So glycine, well, here's our first. We're going to see it again. Glycine, they consider hydrophobic simply because it's not polar, it's not charged, but hydrogen. Glycine in assembling proteins is really used in a place that needs to be flexible, because you don't have a big R group like here that might get in the way of a bend. And we're going to see that proteins are going to bend. So, glycine, alanine, valine, leucine, isoleucine, proline, tryptophan, phenylalanine, methionine. Those are in our hydrophobic category of amino acids.
So, I think you just answered the question. But so by the category, you just mean like it's hydrophobic or hydrophilic?
It's going to be, we're going to have 4 categories. Okay.
But
this one you can say hydrophobic.
Okay.
Yeah.
What is the 1-letter abbreviation?
We're going to get them. Yeah. Well, it's not just the first letter. Some because we have some that have the same first letter, so they're different. That's why it's important to just commit them to memory. So here we have 2 groups. Now these are hydrophilic, and so we have the polar uncharged amino acids. So we've got serine, threonine, cysteine, tyrosine, asparagine, glutamine. And then here we have still polar. These are uncharged. These are polar charged amino acids. So we've got glutamate, lysine, arginine, histidine. I can't make this one out. That's all aspartate. Now, if you're writing all these down, I'm going to make it real easy for you in just a minute. Okay. Come on.
So acidic or basic, is it the positive or negative?
Yeah, positive or negative. So here are our names. Here are our 3-letter abbreviations. 3-letter abbreviations are pretty straightforward. But the single letter abbreviation, see we got 4 A's. Alanine, A. Arginine, whoops. Asparagine, N. Aspartate, where they got the D, I don't know. So you just have to commit these 1-letter. Lysine, K. So, right. You just have to commit those to memory. This one's kind of phenylalanine. This is going to trip you up if you're not careful, because you're going to want to spell the word with an F, because that's its single letter abbreviation, but it's a PH. It just sounds like an F. All right? How we're going to do this. Here's our hydrophobics. And notice again, glycine in this particular illustration, they even slide it into the hydrophilic. You probably could put that right on the edge, since it's just a hydrogen. So we have hydrophobic, hydrophilic, uncharged, and polar. And then we have our charged, which is our acidic over here, which is nice, they put acid with it, and basic over here. So we're going to break them up into those 4 groups. Okay? So this is how you're going to do this as you study for the test. And I see a lot of color pencils. I love color pencils. I love color highlighters. My books look like rainbows. So that looks like a mess, and you might be kind of getting anxious right now thinking about memorizing all this. But I said structures don't matter, right? So let's get rid of structures. Now we got colors. So if we take the first letter abbreviation with the colors, now what do we have to memorize? We got to memorize 20 letters and 4 different colors. You got it. But when you look at everything else, it's like, I will never be able to remember all. See how I like simple? And I like condensation reaction, condensing. See what I did there? Can throw away the water. I like condensing information down because you know most of the names of the amino acids. So you don't have to look at those names. That information is in here. When you study what you are memorizing and working to memorize are ways in which you can recall that information when you need it. And let me encourage you, when you are studying over the weekend, I know that sounds like an oxymoron, studying weekend, but you got to in the summer. As you're studying over the weekend, get away from your notes, get away from your book, get away from Google, get away from flashcards. You have to be able to sit down with a dry erase board or a blank sheet of paper and recall everything you need from right here. Why do you have to do that? That's all you're going to have on the test. And if you have done like many people, spend hours and hours writing out flashcards, and you look at those flashcards, right? Okay, so what is the amino acid that has hydrogen as the side chain? Oh, that's the question. What is the amino acid has hydrogen as a side chain? Oh, I know this one. It starts with a D, a G, oh yeah, glycine. You cheat. And if that's the only way you study, you're going to get that question on the test, and you're going to go, oh, that's that question. Hydrogen is a side chain. And you're going to pick up the test and look on the back, and it's not there. Have you ever done that? Pick up the test and go, oh, I wish it was there. You've got to get away from everything. It's in here, so don't try to remember every word on every page of every PowerPoint slide or every page in your book. Find ways, like I've done here, to condense that information down so you can memorize those colors and those letters, knowing that it's going to help you remember the bigger stuff. That's the whole trick to being successful. And if you memorize those little things like that, or little mnemonics and sayings, you're going to take them with you the rest of your life. And if your career is in the biological sciences, you will use them. And even if you're not going into the biological science, learning them will help you do better on the test. That's what we're worried about, right? That's our immediate future. All right, this is just another chart, again, showing you the amino acids. And if this is helpful to you, great. If not, don't look at it. That's just there for you to study on in your time. So proteins are assembled with a condensation reaction. That's generic. So we're going to specify it a little bit, because with this condensation reaction, we're going to see that we take one amino acid, our monomer, and the second amino acid, and we're going to assemble them together with an enzyme, liberating a molecule of water. See, condensation. And because we're going to take this oxygen off of the carboxyl group and the 2 hydrogens off the amine group, do you see how we're always going to have a directionality? Your protein is always going to have an amino terminus and a carboxy terminus, a positive end and a negative end. And it's always going to be assembled in the same direction. And our genetic code tells us which amino acid is going to come next. And this particular kind of bonding between the carbon and the nitrogen of the carboxyl and the amine is specifically called a peptide bond with amino acids. That's why when you assemble amino acids into a relatively short protein, sometimes it's called a what? Peptide, because of those peptide bonds. So we're going to use energy, we're going to use enzymes that are going to make this happen, and principally this is going to be happening within the enzymatic properties of the ribosome, where we're going to assemble the amino acids into proteins. So again, here's just a little animation showing liberate the water condensation reaction. And there are times we need to break amino acids down, so we use hydrolysis. Comes into our peptide bond, bada boom, carboxyl, amine, to separate them out. But do you see how it's still the generic condensation hydrolysis? We're just applying it specifically to our proteins. So with our proteins, we're going to have different levels of organization. Our genetic code in our DNA dictates which amino acids are going to be assembled in which order. And the order of the amino acids is referred to as our primary sequence. That's our primary structure of our protein. But we're not finished. Just knowing the amino acid sequence of our proteins isn't enough for our protein to be functional and active, because we may have to fold that protein. And two principal structures that are used are called alpha helices or beta sheets. Sometimes they're called beta pleated sheets. Have you seen the pleats of curtains or a skirt? That's sort of the design of our protein. It will have a run this way and then will come back this way, and it makes sort of a layer. Some of those layers may form barrels. But we can have the arrangement of the amino acids that may spiral abound. That is an alpha helix. So whether we're a sheet or a helix, that is our secondary structure. This is going to be dictated by the arrangement of the amino acids and the properties of those amino acids. All right, so primary, the amino acid sequence. Secondary, the folded nature, alpha helices, beta sheets. And then we get to the tertiary structure, which basically is the three-dimensional folding of the entire protein chain. You can see that we have an alpha helix and some other parts to this particular molecule. That's going to be the tertiary or the third level structure. And this particular protein that's being illustrated, this is called a globin chain. And this globin chain has a specific arrangement of amino acids in a very specific location that can bind with that little black dot, which is a molecule of iron. That arrangement of amino acids is called a heme group. So if this is a globin chain that has a heme group that can bind iron, what do you think we call that? Hemoglobin. But this is one of the globin chains of hemoglobin. That's not the hemoglobin molecule, because the hemoglobin molecule is comprised of 4 globin chains, 2 alphas and 2 betas. So when we get the final arrangement of those 4 proteins, the 4 globin chains interacting together as one functional unit, that is our quaternary structure, where we have 2 or more proteins associated together. That is our final functional molecule. So do you see the organization? Amino acid sequence, initial folding, the three-dimensional fold, get it, three-dimensional tertiary folding. Quaternary is just multiple proteins put together, if that's needed to form a fully functional molecule like hemoglobin. Does everyone see those 4 levels? And it's those 4 levels regardless of our protein chain. There we've got a good view of our hemoglobin molecule. The red illustrates the heme group that binds iron, and it's the iron that hangs on to the oxygen, not the protein itself. So how do we fold the proteins? The shape we call conformation. Hey, there's our old friend. We can have hydrogen bonds. We can have ionic bonds. But what we're never going to have, other than the peptide bond, which is a form of covalent bond, by the way, between amino acids for folding, we're never going to have covalent, because covalent's concrete. We're never going to form those. But we can have disulfide, where you have two amino acids that have a sulfur. These are cysteine amino acids. If they get close enough together with those sulfurs are going to lose the oxygen and hydrogen, and they're going to stick to each other. And you see how you have this is referred to as a disulfide bond between two fairly distant amino acids. The antibodies of your body, they have tons of disulfide bonds that help form the quaternary structure of what we know are our antibodies. You'd say those are pretty important, right? Yeah. So hydrogen, ionic. We do have some hydrophobic. If you have enough hydrophobic amino acids in that first orange group, if they're close enough together, you can have some pretty powerful hydrophobic interactions. And where these are going to be important are those proteins that pass all the way through your plasma membrane. Because the part of the protein on the outside is going to be hydrophilic. The part of the protein on the inside toward the cytoplasm, hydrophilic. What do you think about those amino acids that pass right through the middle? What do you think they're going to be? Hydrophobic. So when you sequence a gene, if you see a run maybe of about 7 to 14 hydrophobic amino acids, as a protein biologist or a cell biologist, the first thing you're thinking about that gene and that protein is this must be a transmembrane domain of a membrane protein. Because those amino acids are going to need to be hydrophobic because they're in that Oreo cream hydrophobic environment where all those fatty acid chains are going to be. So again, we see primary structure, just the sequence of our amino acids. There are 3-letter designations. This is showing some of the positions of our disulfide bonds. This is insulin. And when you put these two chains together, what kind of structure would that be? 2 protein chains together? Quaternary. That's the quaternary structure. This is our alpha helix. You see how it spins around? This would be our beta pleated sheet. We have a run of amino acids. You see how it kind of kinks back and forth like that? And then when it gets up here, this is where glycine is important, where you make a 180 bend. You don't want a big side chain in the way, so just the hydrogen allows it to bend, and it kind of comes back this way, and it can kind of go back up. But you see how it's like a flat curtain? That would be our secondary structure with our helix and our sheet. Here's some 3-dimensional single proteins. So that's our 3D structure, our tertiary structure, and then our insulin. That would be our quaternary structure. Let's stop there. We'll pick this up, hit chapter 4 pretty hard. Try to get this material done because chapter 4 is the last chapter for the test on Tuesday. And hopefully this recording won't be as messed up as it was last time.